and come on over. I'd like to welcome and thank Dr. Preethi Sheshadri from the University of College London. And let me let you start. And she'll be speaking on restoration of lysosomal acidification, rescues mitophagy along lysosomal and mitochondrial pathophysiology in GBA1 Parkinson's. Thank you.
Good morning, all of you. Thank you for providing me with an opportunity to share our data here. Before I begin, I would first like to thank my lab, Professor Dushan lab. That's Michael Dushan, and all of the lab members, especially Alicia, Sylvia, Joanna, and Alessia, who helped me with this project. This work was initially funded by Michael J. Fox Foundation, and then we got funding from Parkinson's UK. I would also like to thank the Brain Foundation for providing me with a travel grant. So the work in our lab started about a decade or so ago when Laura Salame studied GBA1 knockout mice neurons, when she saw that not only in the homozygous knockouts, even in the heterozygous knockouts, there was a considerable reduction in the mitochondrial membrane potential. And since then, we have moved on to patient-derived models. And in this work here, I show the data from patient-derived fibroblasts belonging to E326K and N370S mutations, along with IPS-generated dopaminergic neurons, again belonging to the same two mutations, along with at least one isogenic control. So moving on, when we look into the mitochondria in the GBA1 mutant dopaminergic neurons, we see that both in E326K as well as N370S, the mitochondria are considerably swollen, and there is reduced cristae in these organelles. And even in the lysosomes, when we stain the cells with lysosensor yellow-blue, we see that they have impaired lysosomal acidity, where the lysosomes are less acidic as compared to the control cells. So when we also calibrate the pH in the fibroblasts, we saw that both N370S as well as E326K have an increased pH ranging between 4.8 and 5.05, and in controls it's about 4.5 to 4.7. Now, we have done a whole barrage of experiments to look into the mitochondrial and lysosomal dysfunction. We see that with respect to mitochondria, there is reduced mitochondrial membrane potential, decreased oxygen consumption rate, along with oxidized NADH pool. And in the lysosomes, there is accumulation of the lysosomes, increase in lysosomal pH, and decrease in the proteolytic function. And when we look at the autophagy, there is reduced autophagy and mitophagy in the cells. We decided to dig a little deeper into the autophagy part of the story. And again, when we look into the electron micrographs of E326K or the N370S mutants, we can see here that even in the lower magnification, there is a huge accumulation of electron-dense particles as compared to the controls. And when we increase the magnification, you can see that there were autophagic vesicles that was captured at different stages of autophagy. We went on to quantify these autophagic vesicles as per the Nikor et al. Paper, and found that in the GBA1 mutants, there is an increased accumulation of autolysosomes and lysosomes, whereas the autophagosomal number was not much different with respect to the control cells. To check this with mitophagy as well, we saw that when we stained the cells with citrate synthase, LAMP1 and LC3, again, there was not much difference in the mitophagosomal number, whereas there was increased accumulation of mitolysosomes in the GBA1 mutant cells. Now, all this is fine. The question was, why is any of this is happening? And what has it got to do with so many dysfunctions that we see in both lysosomes as well as mitochondria? And that's when we came across this paper from Willem Palm's group, where they show that mTORC1 regulates the lysosomal catabolic activity via the regulation of the VATPase complex. Basically, what they say is in the presence of amino acids, mTORC1 inhibits the complex formation of ATP6V1 domain on which essentially goes and sits onto the V0 domain of ATP6 for easy proton exchange. Now, VATPase or ATP6 is vacuolar ATPase, which is important for maintaining pH in different organelles. And it's more important with lysosomes because VATPase complex is the one that provides protons to maintain the lysosomal pH. What they also saw is that when under amino acid depleted conditions or under autophagy conditions, there is a complete formation of the VATPase complex, and hence autophagy takes place. Of course, all this is in a physiological concept where there is a set amount of mTORC1 that is necessary, that needs to be active on the lysosomes for it to perform its functions. When we looked into our GBA1 mutant neurons, we found that in the untreated cells, when we did the lysosomal enrichment assay, there was indeed increased active mTOR on the lysosomes in E3 to 6K lysosomes. And with respect to the ATP6 components, there was depletion on the ATP6V1 domain components, and there was not much difference between the ATP6V0 in the lysosomes of untreated or the mutant GBA1 mutant cells. Likewise, there's also an increase in LAMP1 that we see. When we then treated them with rapamycin, there is a decrease in the active mTOR levels on the lysosome, and there is a increase in the ATP6V1 components in the lysosomes of E3 to 6K as well as N370S. And there's again not much change between the ATP6V0 levels in the mutants with respect to that of the controls. But we all know how big an elephant mTORC1 can be, given how it is involved in a lot of notorious pathways that mTORC is important for. So but before that, we also sort of wanted to confirm if ATP6V1 is indeed not forming a functional complex, and for which we decided to probe the cells with ATP6V1B2 component, which was tagged with M neon green, and the ATP6V0 with M scarlet, and looked into the fluorescence lifetime of the ATP6V1. So when ATP6V1 is in its unbound form, or it's there in the cytoplasm, the lifetime of ATP6V1 is higher, whereas when it is in the bound form, you would ideally expect that the lifetime is lower. And so when we did the fluorescence lifetime experiments, we indeed saw that the average mean lifetime of ATP6V1B2 was increased in the GBA1 PD cells. And when we treat them with rapamycin, this increase is rescued. Now again, like I said, mTORC1 is a big pathway to modulate, and we don't know what all effects it can have. And to also know if it's indeed pH that is doing something to the cells, we decided to collaborate with Professor Mark Grinstaff from Boston University and Professor Orion Shirihai from UCLA. So these acidic nanoparticles co-localize specifically into the lysosomes, as you can see here with the rhodamine tagged acidic nanoparticles, co-localized with lysis tracker blue. There's not much difference, and the acidic nanoparticles are also able to rescue the lysosomal pH in the cells, in fibroblasts. And when we look into the GBA1 mutant dopaminergic neurons, in the untreated cells, both the E3 to 6K as well as N370S mutants have impaired lysosomal acidity, whereas upon treating with acidic nanoparticles, there is a rescue in the lysosomal acidity in the cells. Likewise, with respect to mitochondria, we stained the cells with TMRM, a mitochondrial membrane potential dye, and both the mutants have reduced mitochondrial membrane potential, and treatment of acidic nanoparticles rescues this property in the cells. Even with respect to mitophagy, we saw that, like I said, there is reduced mitophagy in the GBA1 PD cells, and treatment of acidic nanoparticles was able to rescue the mitophagic property in the cells. And here we have probed the cells with mitochima, which is a ratiometric probe for looking at mitochondria in its acidified or less acidified state. In conclusion, we know that in a healthy condition, almost everything should be perfect with respect to functional mitochondria, functional lysosomes, etc. And under GBA1 Parkinson's disease, there is dysfunctional autophagy and lysosomal pathway with dysfunctional lysosomes and dysfunctional mitochondria. And in our case, when we treat them with acidic nanoparticles, we see that these nanoparticles are able to rescue the pH, improve their proteolytic activity as seen by decurate BSA, along with improving the mitophagy and improving the mitochondrial function with respect to the reduction in the oxidized NADPH pool and improving the mitochondrial membrane potential. Thank you.
Thank you so much. Yeah, we have time for a question. Yes?
Yes.
What attracts the acidic nanoparticles into the lysosome?
So these lysosomes are designed to be specific to the lysosomal into the lysosome. So how it is taken up is by the endocytosis. So it comes via the pathway of the endocytosis, where it goes into the lylate endosome and then comes into the lysosomes.
And then once it's in there, the protein horse liberates the acidic receptor?
Yes, that's how I understand it. The more details of it would be by Mark, who has designed the nanoparticles.
So you're proposing that as a potential?
Yes, I do think we have to look into pH as one of the aspects. Probably, I'm not telling that's the only aspect, but I think probably lysosomal pH is important to degrade all the components that are stuck in the lysosomes because of the impaired pH that is there. And due to this, there's probably an accumulation of mitochondria, which we see in our work. There's an accumulation of mitochondria which are dysfunctional, and so when you degrade when these particles are degraded, the pathway is probably going to go back.
What's known about what's driving the elevated pH within?
The elevated pH is due to this increase in active mTOR, which increase in active mTOR would mean there is impaired complex formation of ATP6V1, the VATPase complex basically, where the ATP6V1 domain is unable to sit onto the ATP6V0 domain on the lysosome. That would be one of them. I wouldn't say that's everything, but because there is some existing pH that is there in the cells. It's just that it is not sufficient for the degradation of so many vesicles that are there in the autolysosomes.
Okay, thank you so much. That was great.